series on genomic science. And to introduce our today's speaker, I request Dr. Nikita.
Good afternoon. Today we have with us guest lecture by Srashti Jyoti Agrawal, a PhD scholar from Delhi, under the guidance of Dr. Bhavana Prasher Madam. Agrawal Madam will be guiding us on the topic of genomic science. I welcome you, ma'am, and request you to start with the lecture.
Hi everyone, I'm Srashti. I am currently doing my PhD in mitochondrial research. So, I basically try to understand the function of different genetic mutations in mitochondrial diseases. Today, we'll be having a lecture on genetic variants. So, in the last lecture, we learned about, sorry, in the last lectures, we learned about the central dogma of life, how DNA gets replicated and then it transcribes to RNA, which then further gets translated to protein. And we also learned the genomic structure, how it is organized into the nucleus, and what are the functions of this organization. So today we will take this a step further and learn about the different variants of genomes that are available. Uh, so like I said, DNA gets transcribed to RNA and RNA gets translated to protein. Now, this RNA to protein translation is possible due to the genetic code. Like we see different algorithms that are in place. Our own DNA also has a specific kind of algorithm, which is the genetic code. So basically, RNA is read as three-letter genetic codons. These codons basically can specify a special amino acid as seen here. And each of these 20 amino acids in the combination can form the protein. So there are 64 codons and different combinations of these codons can form different amino acids. So for example, here you can see this RNA. And if we read it from here, UAC, which forms tyrosine, UCA, which forms serine, then CCU, which forms proline, and UCA, which again forms serine. So here, like this, if it's read three bases at a time, it forms the sequence of amino acid, which is called a peptide. Now, when these three-letter codes are read, this is called a frame. So, if it's read from the first amino acid or the initiation amino acid, it's called the first frame. Then, if it's read from the second base, it's called the second frame. And if it's read from the third base, it's called the third frame. These are basically the possible frames that are available for the genome to be read and translated to protein. So, like for a specific book, there are millions and millions of copies available globally. However, each copy might have uh, some kind of typo or some kind of mistake, maybe. So, this mistake makes these copies different from each other. It might or might not lead to changes in the text which is being read, but it is still typo nonetheless. Similarly, our genome, which is present in all of us, has different typos available. So these typos are called the genetic variants, and these are compared to a reference genome, which is basically a specific set of uh, nucleotides bases, which is considered the reference. So anything that is read in any of us will be in comparison to this reference genome. Now, approximately 0.4% of the human genome is variable to the reference genome, and 99.6% of our genomes are quite same. So, these variations occur due to mutations. And what is the basic difference between mutations and variations? Mutations is a terminology which is specifically used for changes in the DNA when we want to connotate the meaning that the mutation has negative implication. However, these variants, like I said, may or may not lead to change in the text, that is, change in the meaning of how DNA is read. So, therefore, every variant comes under the umbrella term of variant. If this variant is present in more than 1% of the population, then this variant is called polymorphism. So, for example, some of you might have heard the terminology single nucleotide polymorphism or SNP. Now, if this variant is in the frequency between zero point 0.1 to 1%, then this variant is called low frequency variant. And if it's present in less than 0.1% of the population, it's called a rare variant. So there are different types of variants. For example, single nucleotide variations, which are just substitution of a single nucleotide in a specific locus. Then we have indels or insertions and deletions. These are small insertions and deletions which are less than 50 nucleotides in size. Then we have copy number variations or CNVs. These are basically increase or decrease in the number of repeats that are present in our genome. So our genome also has something called short random repeats, which are STRs. These are basically repetitions of the same basis. So if something happens in these bases, if they are increased in size or decreased in size, they are called copy number variations. And then we have structural variations, that is, these mutations, basically insertions, deletions, duplications, which are more than 50 nucleotide in size. So we'll go through them one by one. First, let's talk about single nucleotide variations. So if the single nucleotide variation is happening, that is, one base is changing in an exonic area, and in these exons, if this change is changing the codon as well. So, for example, from AGT, it's becoming AGA, then we call these missense or non-synonymous variations. Earlier, AGT the amino acid for AGT is serine. However, for amino acid for AGA is arginine. So serine is changing to arginine. That is why they are called missense or non-synonymous variants. Then we have synonymous variants. For example, since we have 64 codons and we have 20 amino acids, so there are some codons which code for the same amino acid. For example, here AGT as well as AGC, where the single nucleotide variation is occurring, they both code for serine. Therefore, if the amino acid is not changing even after the variant is present, then we call this mutation synonymous. If the mutation is happening in non-exonic areas, for example, regulatory regions, then they do not fall into the exonic regions. Therefore, they do not fall under the synonymous, non-synonymous category. They are regulatory mutations. Then we have nonsense or stop-gain mutations. Basically, there are three codons which specify stop codons. So they tell the polymerase where to stop translation. So when the polymerase encounter the translation gets stopped, it gets halted there. So basically the protein does not form after that. The peptide doesn't extend after that. So for example, here TCA became TGA after changing this C from G. Mutation happened in this C from G. And here instead of serine, it got to become a stop codon. So therefore, the peptide sequence would stop here and this whole sequence would not be translated. So these are called nonsense or stop gain mutations. Then if the variant might still occur in exonic region or it might occur in intronic region, but sometimes these variants affect how splicing occurs. Now, splicing is important for pre-mRNA to become a mature mRNA. And if it's not, if it does not occur properly, then proper splicing would not take place and a complete uh, mature mRNA would not form. So if, for example, here a mutation occurs in this exon-intron boundary and in the motif that defines the splicing, where the splicing occurs, then it might lead to either intron retention in the mature mRNA or exon skipping in the mature mRNA. And this might lead to shift of the frame or incomplete protein forming, or maybe just insertion of stop codon. So these are called spliced effects. Next, we will talk about the insertions and deletions. So when insertions and deletions of less than 50 nucleotides happens, these are called small indels. So depending upon where these indels are happening, the functional implication of these mutations could be different. For example, if it's happening in the motif that defines the splicing area, then it might lead to spliced effects. If it is happening in some place and it is leading to shift of the frame and thus a stop codon insertion, then it would be stop gain. It might lead to frame shift also, and most common small indels lead to frame shift mutations. Or if the mutation occurs, that is insertion of deletions in the multiples of 3 happens, then it might lead to inframed indels. Because since codon is made of 3 nucleotides, that is why the frame of 3 matters here. So if the insertions or deletions happen in the frames of 3, then inframed indels occur. For example, here we can see that there is a deletion of these 5 nucleotides and which has led to shift of the frame. After this alanine, we can see that instead of this threonine, we can see that there is a shift of the frame, and we can see that serine, and lysine got incorporated. Also, the length of the protein got affected. So this is what typically happens in the case of deletions if it happens in the exonic area. Then in the case of insertion, as you can see in this area, there is an insertion of 4 bases. So since there is an insertion of 4 bases, there would be a frame shift mutation occurring here. And the size of the protein increases and also the frame changes. For example, after threonine, histidine, glutamine, and lysine should have been there. However, now it's lysine, threonine, serine, and again lysine. So this shows how small indels can bring about the change in the frame of the translation. And this now imagine that this mutation occurs in an area which is very important for the protein. So for example, a catalytic domain or a structural domain, the C-terminal domain. And then because of that, the domain does not form properly. And due to which the function of the protein gets affected. So therefore, these mutations are very deleterious if they happen in the exonic area. Next, we will talk about the copy number variations and the structural variations. So, like I said, there are short tandem repeats present in a genome. For example, here we have TCT, repeats of the TCT. And if the repeat region, maybe in the repeat region, maybe deletion or insertion happens of the same copies of the same basis. For example, here T into TCT gets deleted and we only have one TCT repeat left. So therefore, there is a deletion of 3 amino acids here. And this is how copy number variations function. So this might or might not happen in a frame specific manner. And these also might lead to frame shift mutations or in frame mutations. Then we have structural variations. Now, like I said, if deletions, insertions, or duplications, they happen for more than 50 nucleotides, then they are termed as structural variations. Uh, it might happen that a few of the bases, a few thousand bases, maybe a few 100,000 bases got deleted. And this might lead to deletion of one gene or one domain of the protein, or maybe multiple genes in many cases. Again, in duplication, this might lead to insertion of a certain specific area, and this might affect the expression of a specific gene or multiple gene. Similarly, in inversion, it might lead to, for example, a specific area of the genome got inverted, and its configuration got inverted. So, for example, if it was in this direction, it might go to the other direction in the other after the inversion, and this might affect the regulation and expression of the genes. Then we have insertion. So basically, in insertion, a large chunk of DNA gets inserted where it should not have been inserted, and it might affect the expression of the protein or multiple genes, or it might also lead to structural changes in the chromosome. Then we have translocation, that is a specific area of one chromosome gets transferred to the other chromosome, and uh, this can happen in both ways. For example, it can get translocated from here to here, and this translocation from this chromosome will also give its part from here to here, or it might happen that just this area gets translocated to the other chromosome, and this is very deleterious and it might lead to certain diseases. So we learned about the structural variants and other variants, but how do these variants arise? There are multiple mechanisms which might lead to the genetic variants. For example, errors in replication, recombination, or maybe insertion of mobile elements, or errors in DNA repair, introduction of specific mutagens, or just in general the cellular stress. So, uh, like I said, basic uh, processes of our own body can also lead to these variants if they are not kept in check. So, for example, in replication, we have two strands of DNA, and then these strands open up to make sure the polymerase can sit over here, and then replication can start at the replication fork. So this replication happens for each of these two strands. One is called the leading strand, in which the replication occurs from 5' to 3' direction. And the other is called the lagging strand, in which the replication happens from 5' to 3' direction. But since it has to start from the opposite direction, it creates small Okazaki fragments, which are small fragments of DNA, which are then joined later. So this is how the replication happens. The fork keep moving in the direction of 5' to 3' of the new DNA, and thus we get the replicated DNA, and the two strands of it. So if there are any errors in these replication, then mutations would be incorporated while uh, replication is happening. So there are multiple types of DNA polymerases which are required for different functions during the replications. And there are also other proteins which are required. For example, helicases, topoisomerases, primases for different functions. And for each polymerase, there is a specific fidelity which is defined as the ability of the uh, polymerase to make sure that no mistakes occur. So high fidelity means that there would be fewer mistakes while replicating, and low fidelity means that there would be more chances of mutations being incorporated. So this fidelity is maintained by mechanisms such as the proofreading quality of the polymerases or other proteins, the repair or base selection quality of the polymerase. So just like when you are copying from somewhere and you check once before you're submitting the paper or submitting the whatever you are copying. You check once whether you have made any mistakes, just to make sure that you have not incorporated any errors. This is called proofreading, and this is what the polymerases also do. So the basic major polymerases that are involved in DNA replication, eukaryotic DNA replications are Pol alpha, Pol delta, Pol epsilon, and Pol gamma. And as you can see, each of these have a specific role, and each of these have different fidelity. For example, Pol alpha is involved in primer synthesis. It synthesizes the primer so that the replication can start. Since it has to only create primers for of only a few bases, therefore it does not need that much proofreading ability, and it has therefore low or no proofreading ability. However, Paul delta and Paul epsilon are involved in leading and lagging strand synthesis, and therefore they need to have high proofreading ability so that they can make sure that in the new strands that they are synthesizing, there are no errors. Similarly, Paul gamma, which is important for mitochondrial DNA replication and is the major polymerase that gets translocated to uh, mitochondria and performs DNA replications for mitochondria. It needs to have high proofreading so that there are no mutations in the mitochondrial DNA. Similarly, if there are errors in the recombination mechanism, which is an evolutionary mechanism to make sure there are genetic variations in the genome available for us. Uh, therefore, if there are any errors during the recombination, for example, errors in recombinations might lead to translocation, or structural variations may be large deletions or insertions. So therefore, errors in recombination can lead to large structural variations, and it is very important to maintain proper recombination in the cell. Then we have insertions of mobile elements. These mobile elements are basically jumping DNA, which can jump from one place in the genome to another. And evolutionarily, it has many functions. But in humans, mostly retrotransposons are currently functional, and they copy and paste via RNA intermediate, that is reverse transcription. For example, lines, signs, or like allorepeats and LTRs. So their function is to increase the copy number if it goes lower. But if they get inserted in a region where they should not have been inserted and might hinder with the gene's function, then this might lead to a deleterious mutation. Then we have the DNA repair pathway. Normally, all the errors that are performed that take place in the cell while replication or while division, they are taken care of the DNA repair machinery. But sometimes some of the errors are not taken care of, and hence, if there are any errors during the DNA repair, they might lead to sustenance of these mutations in the next strand of DNA which has been synthesized. They are most majorly of three types. Single strand damage repair, in which only one strand of the DNA double helix would be modified to take care of the repair pathway. We have double strand break repair, in which both of the strands would have to be modified to take care of the repair. And then we have specialized repair, which are only applicable in if certain uh, case occurs in the cell. So, in the single strand repair, we have base excision repair, nucleotide excision repair, and mismatch repair. So, DNA is formed from these four bases: thymine, adenine, cytosine, and guanine. And in base excision repair, uh, the base from the sugar diphosphate backbone is maintained as such. However, the bases are first excised, and then a new base is put in its place to take care of the error that has been here in this base. In the nucleotide excision repair, this whole nucleotide is excised along with the phosphodiester sugar backbone, and base excision repair usually happens for only one base. However, nucleotide excision repair can take place in multiple bases in the same time. Then we have mismatch repair, which takes place after the replication has occurred on the newly synthesized strands. So wherever the repair machinery mismatch repair machinery feels like there are lesions or bulges occurring after replication, it corrects the base on the wrong strand wherever the mismatch is present. So for example, the correct matching of thymine is with adenine. However, somehow thymine is incorporated with guanine on the complementary strand. So mismatch repair machinery would recognize this as an error and would correct it so to make sure that thymine base pairs with adenine correctly. Then we have double strand repair. These are majorly of three types. Homology directed repair, in which the machinery checks the regions in the other strand which are homologous to the strand in which error is present, and then it creates a template and it fills in the gaps wherever there are gaps. Then we have non-homologous end joining, where there is no repair templates, just random bases are put in between, hoping that the other repair machinery would take care of it further after the replication. Then we have microhomology mediated end joining, in which small areas of homology, which are called microhomology, which are from 5 to 25 base pair, they are searched near the error. And these when the microhomology regions are found, then it's made sure that the end resection happens. That is, the ends are staggered ends are created, which are also homologous to each other, so that the ends can anneal and DNA synthesis can occur and thus take care of the gaps that are present in the DNA. These double strand repair is very important because if replication fork in the next cycle or in the same cycle, it is recognized, then the replication fork gets stalled and it might be deleterious for the cell because the stress response is activated. That is why in the double strand repair, accuracy does not matter that much as to repair the double strand, and then the other repair machinery can take care of the rest. Then we have if there are specific mutagens. Mutagens are compounds which can trigger the DNA to change itself, uh, and there are three major types of mutagens: chemical mutagens, physical mutagens, and biological mutagens. So, chemical mutagens are either they are base analogs, for example, 5-bromouracil, or they are intercalating reagents like ETBR, or they are metal ions or alkylating agents. So they basically interfere with the DNA replication or DNA repair machinery. Then we have physical mutagens like heat or radiation, which can change the uh, change the bases in the DNA, which would then be recognized in a different manner in the next replication cycle, and thus leading to errors. Then we have biological mutagens like virus, bacteria, and transposons, which might lead to either changing of specific bases or insertions of specific elements, thus leading to different type of mutations. Also, cells' own cellular stress can trigger the response. For example, if there are too many reactive oxygen species in the cell from mitochondrial respiration or peroxisomes or due to inflammation, then it might trigger oxidation of nucleotide bases and create abasic sites or single strand breaks or base modifications, thus leading to changes in the DNA, and which might or might not be repaired depending upon the repair machinery. So these variants, if they occur in the somatic cells, then they are called the somatic variants. But if they occur in the germ line, that is either the sperm or ovum, then they would get transferred to the next generation, and therefore they are called the germline variants. So Somatic variants might also lead to specific deleterious effects, but mostly, if the germline variant is deleterious, it might be hazardous for the next generation. So now, on this line only, we will try to understand why is it important to study these variants. From a research perspective, studying these variants can give us insights into functions of gene, regulatory mechanisms that are occurring um, in a specific cell or a specific organism. For example, if we want to understand how cancer is triggered in a specific cell, we can create a specific mutation leading to introduction of a cancerous state. Or if we want to understand the function of the gene, we can create these mutations in a model system like mouse or rat, and we can understand the function of the gene depending upon the mutation that is there. Then they have a lot of genetic variants also have a lot of implications in evolution and lineage checking. So, for example, if each one of us looks different, and that is possible because all of us have genetic variants present in us. So, that is why a specific component of a genome is different, and which makes us different from each other. So, from that perspective, it is important for genetic evolution. Also, to understand the conservation of specific proteins or protein domains or genes in general, to understand how they are conserved from our ancestral species to us, it is important to understand certain genetic variations or to understand the familial lineage. For example, if we want to know which uh, race we are from or which kind of um, group we belong to. These genetic variants can help us understand the lineage as well. And also, they are important for in the forensic science as well because, for example, DNA fingerprinting is an technique which is possible only because there are genetic variations in each of us present. Now, from a clinical perspective, uh, since there are many genetic variations which can lead to diseases, therefore, from a clinical perspective, these variations can also give us insights into disease diagnosis or therapy, uh, or maybe these variations can act as biomarkers. For example, in specific cancers, if a specific mutation is present, we can say that this might be a special type of cancer. So this is how genetic variants can act like biomarkers. So what can we do regarding these genetic variants from a clinical perspective? So first of all, we want to know how to detect these variants. So there are different techniques for detecting these variants. First and foremost, we have Sanger sequencing, which is the first generation of variant detection method, in which the DNA is read by amplifying the DNA in the sample. Then we have next generation sequencing, in which either a specific gene panel of genes or the whole exome or the whole genome is sequenced. We also have array-based methods in which probes are generated depending upon the variants already known to us, and we can understand which variations are present in which individuals depending upon either single nucleotide variant arrays or comparative genome hybridization arrays. Each of these methods have their own benefits and their own disadvantages, and they are all used depending upon uh, what we want a result to be. For example, if we want to catch structural variants, we would either perform whole genome sequencing or comparative genome hybridization arrays, or maybe long read sequencing. If we want to catch a gene, if we want to catch a very disease specific mutation, we would either go for Sanger sequencing or targeted panel or SNP arrays. So each of these methods have their own advantages and are used in specific situations. Now, after we have reached the genetic variant, we want to understand what that variant is. because as you will learn in your databases lecture, there are certain types of files and certain types of outputs that we get from each of these um, techniques. So that output needs to be analyzed, and there are specific variant annotation softwares like Ensemble VEP or ANOVAR, which gives us detail about the variant as well as the gene it is present in, or the specific nomenclature for that variant, the frequencies present um, in the publicly available databases, how frequent it is present in the population, or the effect it might have. Is it nonsense? Is it stop? Is it synonymous? Is it non-synonymous? Or the computational predictions. That is, if there are certain algorithms which are already present in the publicly available resources, whether they predict this variant to be pathogenic or deleterious or benign, that it might not do anything, or existing reports for that variant, whether that variant is already reported to be causing the disease or whether it is reported to be not causing the disease, whether it is very benign. So, depending upon the annotations, we can classify these variants as pathogenic, likely pathogenic, variant of uncertain significance, that is, we still do not know, we still do not have enough information about this variant, or likely benign or benign. So, depending upon whether the variant is in which category, the diagnosis can be given. Now, this ACMG and AMP guidelines, which can classify these variants into 5 categories, this is only performed for genetic variants which are disease associated. And for different diseases, for different types of diseases, the guidelines differ from each other. So, for example, for somatic variants, the guidelines are different. For mitochondrial variants, the guidelines are different. But the uh, the basis remains the same, that we annotate the variants and gain information about the population frequencies, the computational predictions, and the variant type, uh, the functional data, the segregation data from the literature, and these kind of information is collected, and then we classify this variant according to different criteria that are given for each of these annotations. Now, let's go a little bit deeper into the genetics and inheritance of these genetic variants. So, first of all, if a person has a specific genetic variant, uh, and we want to refer to that genetic variant or that specific variant, we would call it a genotype. That is, the different varieties of heritable characters for a gene, for the gene in question present in an individual. For example, in gene A, mutation B is present. So then the genotype would be A.B. So there are specific nomenclatures present for this, which given by HGBS, which are called HGBS IDs. Then we have the phenotype, that is the cumulative effect of that genotype or the heritable characters, which are usually present in an individual. So we can, whatever we can visually see as an effect of that genotype is called the phenotype. Then we have inheritance of characteristics from parent to progeny. So, like I said, these genetic variants, if they happen in germline, they can be inherited from parent to progeny while they are passing on the DNA material. And different varieties of these heritable characters, they are called alleles. So, for example, we have 2 copies of the DNA. In copy A, we have, in first copy, we have allele A. In the second copy, we have allele B. So we might have different alleles depending upon what we gained from our parents. So one allele is passed on from each parent, and uh, depending upon at a specific locus, what genotype is present, we can say that we can see what allele is present. So these variants are inherited uh, either in a familial manner or in a de novo manner. So, for example, if in a cross of mother and father. The father was heterozygous for a mutation, then there is a 25% chance that the progeny would be heterozygous as well. If the progeny got that mutation from the father, and the father as well as the progeny had that mutation in all the cells of their body, this is called familial germline mutation. Then, if the mutation occurred in the germ cells, but the father did not have the mutation in all the cells of his body, then uh, the progeny would have the mutation, but it would not be familial, and this would be called de novo mutation. Then the third scenario is when a few of the cells of either the father, the few of the cells of the progeny contain a specific mutation. So it is not necessarily, it might or might not be present in the germline, but not necessarily present in the germline, and it is present in multiple cells of the body, but not all cells. So this type of individual is called mosaic, and these kind of variations which arise in the somatic cells are called somatic variants. So depending upon the alleles that are present on a specific locus, we can term this genotype as either homozygous or heterozygous. So, for example, here consider the allele B at this locus. If both the strands have this capital B, then we have homozygous BB. Uh, Here, capital B being the dominant allele and small b being the recessive allele. If we have on one strand capital B and on the other strand small b, then we have heterozygous genotype. And if we have on the both strands small b, we will have the homozygous small b genotype. So depending upon whether this capital B is dominant or small b is recessive, the phenotype would be visible. Other scenario is if allele from only one chromosome is present in the progeny. This happens in case of sex chromosomes. For example, we only receive one X chromosomes. For example, men have only one X chromosome and one Y chromosome. In that case, the mutation can be hemizygous because there is no other chromosome to have the mutation. There is only one locus. Then we have mosaics, like I said, alternate allele is present inconsistently in the genome. Here, alternate allele is the variant allele. Then we have compound heterozygous, that is, if there are different alleles present at other locus. For example, here in the same gene, we have capital B as well as capital C, either in the same chromosome or the other chromosome. So this is called a compound heterozygous scenario. So to make it easier for us to understand the genotypes and how they are segregated in across generations, pedigree symbols are generated and pedigrees are created for the families. So each symbol means a separate. Each symbol has a separate meaning. For example, a square represents a male. An unfilled circle represents a female. For mating, we join the circle and square, and then lines from this circle and square represent the progeny. So, if the inheritance is autosomal recessive, that is, the mutation was in the 22 autosomal pairs, and the inheritance would be autosomal recessive because the alleles that are present, they act as dominant and recessive alleles. Then, then the inheritance is autosomal recessive. So basically, autosomal recessive is when you have a carrier person that is having the mutation but does not manifest the disease because there is one copy of gene present there, and one one good copy is enough for the gene to function properly. So even if the other copy is mutated, for example, small d here, So even if the other copy is mutated, it would not matter. It would only matter in the cases where both of the copies are mutated in the genome. So, for example, sickle cell anemia is a disease where is a disease which follows autosomal recessive inheritance. And here you can see the father does not carry the mutation, but the mother does. And again, there is a 25% chance that any of the progeny could have the mutation. And here, for example, the father has the mutation, and the person he's mating with also has the mutation. And therefore, the probability increases. And there is a 25% chance that in the next progeny, there is a person who is affected. So, autosomal recessive inheritance has this characteristic that it skips in generations if the other person also if the other person does not uh, have the mutation. For example, if this person was to mate with a female unaffected female, then the progeny uh, would uh, would be unaffected. Then we have autosomal dominant inheritance, in which having one correct gene is not enough for the function of that gene. It is both the gene, proper function of both the copies are needed for the gene to function properly. Here in autosomal dominant inheritance, there is no generation that is skipped, and the probability of the progeny getting affected also increases. For example, here this male is affected, and approximately 50% of the progeny is also affected, and which is the same case for the progeny of this first generation also. Then we have linked inheritance. Uh, so if the mutation is not present in the autosomes and is present in either X or Y chromosome, then either X-linked recessive, X-linked dominant, or Y-linked inheritance can occur. For example, hemophilia, which is an X-linked recessive disease. And you can see, even though this female had one uh, copy mutated, she is still not manifesting the disease. However, with just one affected copy, this male is showing the phenotype of hemophilia. So even in this, the males have higher probability of manifesting the disease because they only get one copy of X chromosome, and that is also from their mother. So if the mother has one copy mutated, then there is a 50% chance that the progeny male would be affected. Then we have maternal inheritance. Like I told you in the last class, mitochondria have their own genome, and this genome is only inherited from the mothers. So if there are mutations which are deleterious in the mothers, uh, this might transfer in a specific fashion to the ovum, and then the progeny might manifest the disease depending upon the heteroplasmy. So in this pedigree, you can see that the mother is affected and she has the mutation, and therefore, depending upon how many affected mitochondria are transferred to the progeny would also be affected. However, here, since a male was affected and male's mitochondria do not get transferred to the progeny, therefore the progeny of this affected male would be healthy. However, for this female, the progeny would not necessarily be healthy. So the disease manifestation here does not depend upon autosomal recessive or autosomal dominant or things like that. It depends upon the heteroplasmy. Heteroplasmy is the basically the load of variant DNA present in the mitochondria. Now consider this specific cell where there are normal DNA, which are normal mitochondrial DNA, which are depicted in blue, and variant mitochondrial DNA, which are depicted in red. While this oocyte is dividing, the bottleneck might occur, and it might happen that the cell might receive all normal DNA or more high mutant to wild type DNA or high wild type to mutant DNA. Now, depending upon which configuration or which how much heteroplasmy is there, that is how much mutant load is there, it might lead to the disease. Also, a further complexity that is added in mitochondrial disorders is that for each type of mitochondrial DNA disease, the heteroplasmy levels which are required for the disease to get triggered is different. So therefore, the normal Mendelian inheritance does not get followed in the in the mitochondrial DNA's case. So just to summarize, we have single nucleotide variations which can be synonymous, non-synonymous, stop gain, or splice defects. We have insertions and deletions, which can be frame shift, in frame, splice defects, or stop gain. We have copy number variations, which might lead to protein defects or regulatory region defects. And we have structural variations, which might lead to single or multiple gene defects or the defects in regulatory regions. So I hope this has given you a fair idea of genetic variants, and I'm open to any questions.
Participants are requested to write the questions in the chat box if there are any.
I think, Srashti, if I can just come in, Ayagoa, it's a very good presentation, very clear. Have you also kind of written down the further?
Sorry, ma'am, you're on mute right
Ayagoa, please let me in. Yeah.
Yes, madam.
Yeah, so I think the lecture was quite comprehensive. Just wanted to tell Srishti that if she can put the more reading material or any textbook material in the classroom as she did last time, and also write few maybe questions which participants can attempt to answer so that it becomes a revision for them.
now. Sure, ma'am. If there are any questions regarding any of the topics that are covered in this presentation, if you want me to repeat it, I can do that as well. So just let me know in the chat.
The genetic variants, I think a lot of clarity of definitions and ideas, because these people, students especially, must be reading papers here and now then, and they also get to
Yeah.
know the places to go for, and also differentiate between different kinds of variants and accordingly interpret the things. I think that would be for students especially. I think you must learn about the genetic code so that you can always imagine if this change has happened, what could it lead to in terms of amino acid change and stuff like that.
A lot of reading material would be required because there are a lot of topics in this presentation, in this one
Especially
topic.
last part, the inheritance pattern, the pedigree
Yeah,
needing,
that needs to be
yeah,
uh,
practiced
specified
basically.
more.
Yeah, specified more and done more with a lot of examples.
Yeah, true, true. Ma'am, you're on mute right now.
I know, but I'm just waiting for if there are any questions coming up. Otherwise. Typically, these variants are studied for their clinical implications in biomedical research, where you compare. Conventionally, we do case control. Earlier, there used to be family-based studies to identify if any genetic variation is running in the family to be attributed to any quantitative trait. Now there are case control studies where healthy individuals are compared to the any disease of interest, and these known genetic variations are first looked into whether any of these variations are present more frequently in a disease population than the age, gender, and background controlled healthy individuals. So now, even though the number of variants are few, but even if they have a very strong effect, they are likely to be affecting the disease development rate, and also indicating towards disease susceptibility as well as differential drug response. So if one were to study the drug response related variants in a population, for example, if we want to study that warfarin like drug, which has a lot of inter-individual variability in response to drug, or cancer examples that you give, and people typically stratify the population into responders versus non-responders and look for the genetic differences between these groups, and identify, and then also do further validations to attribute that kind of a phenotypic trait to that genetic variation. So these are the kind of clinical applications that people can think of. And then there is a disease susceptibility related studies also done. These are done either cross-sectionally, or people follow it for a long period of time in a longitudinal cohort, and then see who are the ones, in spite of all other background things common, are developing a disease, what are the risk factors, if there are any genetic determinants. So these studies are typically taken either in longitudinal studies or in the cross-sectional studies to assign the clinical significance to the genetic variants. Like, for example, in our first study where we have reported that individuals of extreme constitution types, like extreme Pitta versus extreme Kapha versus extreme Vata, is there any genetic variation which makes them differ? So one of the gene that we have reported, we have found genetic variations in quite a few genes, but one of the gene, EGLN1 gene, where we have found that in the first intron of the EGLN1 gene, at one position, at two positions, there are variations between Pitta and Kapha. So the frequency of that genetic that allele in Pitta individuals is lower than in Kapha individuals, and same way for the other genotype. Now the question was, okay, Pitta and Kapha are differing at that particular position in terms of frequency of that allele to be differing between Pitta and Kapha. Then we went on to find out whether this variation will have any implication on the physiology or on the response to stress, because it is an oxygen sensor gene. So this is how first you identify if there is any difference, and then you try to assign any functional association to it. That is how these variations become important, as then it becomes important as a biomarker or a differentiator or susceptibility biomarker or a response biomarker. I think the lecture was so clear that participants have found it very kind of, interesting and useful. Then more reading will be followed up in the classroom material.
Yes, madam. I think we can continue our discussion in the Google Classroom.
Yes.
So it was really very comprehensive lecture and with so many important examples.
Thank you.
So shall we conclude today?
Uh, yes, madam, we can.
Okay. So thank you very
Thank you.
much.
Thank you so much, Bhavana madam, and thank you so much, Srashti ji. Thank you so much.
Thank you.